Dear participants, I am simply pleased to welcome you to the International Conference on Sustainable Futures, Environmental, Technological, Social and Economic Matters. It is a peer-reviewed international conference, which is the premier interdisciplinary forum for social scientists, life scientists, engineers, and participants to present their latest research results, ideas, developments, and applications. The ICSF occupies contribution in all aspects of sustainable development, focused on the intersection of sustainability, environmental, and technology, and their large implications for corporations, government, education institutions, regions, and social, both at present and in the future. Today, we gathered to discuss workshops biodiversity and ecosystem sustainability. In total, we will discuss 17 reports on different topics. I wish everyone a good mood and safe day. Please adhere to regulation of reports and discussions according to the conference program. In the event that there are technical problems during the work or we go beyond the regulation, please write all questions to the text chat and continue the discussion. I invite Hanna Tutova to speak with a report the topic, the ecomorphic aspect of the soil microfauna community transformation and the recreational impact. One moment.
Soil microfauna community transformation under recreational impact. Soil has natural protective properties. Therefore, even under conditions of intensive anthropogenic impact, the soil microfauna maintains a high level of abundance and diversity. Urban parks perform a wide range of ecological functions, many of which are due to the activity of soil macrofauna. Assessment of the transformation of soil macrofauna under the influence of recreation is important for the creation of effective urban environmental management strategies and environmental monitoring systems. The aim of the study is to identify the main trends in the transformation of the soil macrofauna community under the influence of recreation and interpret them using the ecomorphic approach. Soil macrofauna were collected under conditions of varying levels of recreational exposure. In addition to soil animals, soil properties were measured and vegetation was described. The results obtained on the community structure were subjected to ordination. Some ordination axes were strongly correlated with distance from walkways, indicating their response to recreational pressure. The various aspects of variation in the soil microfauna community are related to the restructuring of the ecomorphic structure. Research aim and objectives. An important scientific problem is to investigate the possibility of applying the ecomorphic approach to assess the impact of recreation on soil biota. We tested the following hypotheses. 1. The community of soil macrofauna in the recreation zone is organized due to the relationship with environmental properties and the structuring of ecological niches of soil animals. 2. The ecomorphs of soil animals are informative and important characteristics of the ecological properties of the macrofauna, which can explain certain aspects of the structuring of the soil animal communities under recreational conditions. Model polygons of public green spaces in Melitopol. The study was carried out on the territory of Park Novoalexandrivskoye. We laid a polygon with a moderate level of recreational load with an average distance to the spontaneous walkways of 4.1 meter. Standard deviation was 3.2 meters. The polygon was a set of 105 sampling points, which were collected along 7 transects placed in parallel, with 15 sampling points in each transect. The distance between the closest transects was 3 meters and 3 meters was the distance between the closest sampling points in the transect. Thus, the sampling points represent a regular grid with a lag of 3 meters of 7 times 15 sampling points, 21 times 45 meters. 35 soil animal species were found in the study area. The abundance of the soil macrofauna community was 493. 10 Indiana/M2. The earthworms were the most abundant and diverse group of saprophagus within the study area and were represented by 3 species. The abundance of the earthworm community was 73. 18% of the total macrofaunal abundance. The endogic species Aporectidia trapezoides dominated the abundance of earthworms. The population abundance of this species was 292.27 Indiana/M2. Together with this species, Aporectidia rosea was also represented by endogic earthworms, and Dendrobina nasinovi was represented by anisic earthworms. The abundance of earthworm cocoons was 23.31 Indiana/M2. The hygromorphs of earthworms were represented by Xerophilus and Mesophilus species. Among the earthworms there are pratons and stepants. The basis of the xenomorphic structure of soil macrofauna in terms of the number of species was represented by sylvants, 47.1%, and pratons, 23.5%. The proportion of stepants, 26%, and paludants, 8%, was somewhat lower. By abundance the situation was somewhat different. The basis of the xenomorphic structure of the macrofauna was represented by pratons, 64.0% in abundance, slightly fewer stepants, 19.0%, and sylvants, 16.8%, and occasionally by paludants, 0.2%. Projections of ecological niches of soil macrofauna species on OMI1 and OMI2 axes. Lower part has negative axis values, upper part has positive axis values. The vast majority of the species on the above lists should be noted to be litter-dwelling species, but at this stage of the analysis it is very difficult to come up with a hypothesis that would explain the observed ordination of species in the community. The RLQ analysis found that 91.24% of the total variation, total inertia, described the first two RLQ axes, 84, 19 and 7.05%, respectively. The Rantist procedure confirmed the significance of the RLQ analysis results at a peer level of 0.017. Conclusion: the ecomorphic organization makes it possible to interpret trends in the variability of the soil macrofauna community under conditions of recreational pressure, and to explain the spatial patterns of the soil macrofauna. The ecological space of ecosystems under recreational conditions is structured between ecological niches of the soil macrofauna. The marginality, tolerance and residual tolerance quantify the position of ecological niches in the ecological space. The variability and soil properties, which is induced by the natural factors or recreation, is a driver for structuring the ecological space of soil macrofauna. The soil animals are the most sensitive to variability and the soil penetration resistance, soil moisture and electrical conductivity. There is a relationship between environmental factors in public green spaces, the structure of the soil macrofauna community and its ecomorphic organization. The ecomorphic aspect of the structure of the soil macrofauna community is more sensitive to recreational pressure than the distribution of ecological niches between species in the ecological space, which suggests that the restructuring of the ecomorphic community structure is a condition for the stability of its organization. The natural variability of soil conditions within the ecosystem manifests itself in changes in the ratio of xerophiles, phytophages, carbonatophiles on the one hand and pratons, paludans and hypercarbonatophiles on the other. A lower level of recreational load is favorable for saprophagus and decarbonatophiles. The zoophages, hemiaerophobes and megatrophoxenomorphs are tolerant to high levels of recreational load. The area that corresponds to the highest level of the recreational load is vacant. This indicates that the most transformed areas are randomly inhabited by representatives of different ecological groups.
Thank you. Are you question? No question. Okay, good. Next speaker. Next speaker, Andrei Igorevich Sidorenko from Bogdan Khmelnytsky Melitopol State Pedagogical University, and topic: biocyanotic influence of the great cormorant in the Azov Black Sea region of Ukraine. I will start presentation.
my report is a biocynotic influence of the great cormorant Phlacocorax carba in the Azov Black Sea region of Ukraine. Large colonies of birds play an important role in soil formation, bringing a large amount of organic matter with their waste products, mainly guano. Special ornithogenic soils with a high content of phosphorus, nitrogen, potassium, and zinc are formed in places where the birds gather. The participation of fish-eating birds in the transport of biogenic substances from aquatic ecosystems to terrestrial ones is indisputable, especially the great cormorant. These birds feed on fish in freshwater bodies, and nests are built on trees in the forest. In nascent places, trees die, and in the territories of former cormorant settlements, the increased content of nitrogen in litter, soil, and plants remains for a long time. Another important type of impact of great cormorant is its relationship with other species of birds, especially the wetland complex birds. Researches of ecological impact of great cormorant were conducted in the Azov Black Sea region of Ukraine in 22 nesting colonies of this species in the period from 2012 to 2021. Most of the expeditional work was carried out using the methods of fixed vehicle, boat, and pedestrian census. Cormorants are most destructive to vegetation during the period when they are building their nest. Bird collects nesting material within a radius of up to 2 kilometers from the colony. Some birds find the material in the immediate vicinity of the nest, others collect it on the neighboring island. Some steal it from the nest of other cormorants. At the locations of the colonies, there is a change in vegetation cover, while in the first year, as a result of strong nitrification of the substrate, species that prefer nitrogen-rich soil prevail. In the following years, even in the absence of birds, the vegetation cover is transformed, including the improvement of the zoonotic and floristic structure of the island complexes. The largest arboreal colonies of great cormorants are located on the coast of the Sea of Azov, the Kazantip Peninsula, Obetychna Spit, the Dnipro reservoirs, Kakhovka, Kremenchuk, Kaniv, Kyiv reservoirs. Everywhere the negative impact of the nesting of these species, both on herbaceous and tree shrub vegetation, is noted, which is the case of large colonies acquires at a significant scale. Coastal plantains suffer the most from the settlements of the great cormorant, as they are chosen by the birds for placing their nest in the first place. Tall Salix alba and Populus sp trees with a good view and an uncomplicated approach to the nest are chosen as nesting biotopes. Nests, as a rule, are arranged in the upper and middle parts of the crown of trees, at the base of middle part of branches of the second order. The negative impact of the growth and development of trees is primarily associated with the impact of bird faces with a high concentration of nitrogen, phosphorus, and potassium on the vegetative organs of plants during the entire nesting period. This leads to the death of leaves and young shoots as a result of burns. In the first year of the appearance of nests on trees, the leaves of the upper and partly the middle part of the crown burn. As a result, the skeletal branches this peels off and the tree begins to dry out. The next year, the upper dry parts of the trees are used by the cormorant as a roost, and the nests are moved to the middle part of the crown. But the end of the growing season, the leaves and 1 year shoots in the middle and lower part of the crown burn. The bark of the perennial shoots peels off and the tree dries up completely. In the future, dead trees are used by cormorant as roost and nests are moved to the nearby living trees. In the absence of trees, cormorant begin to use shrubs as nesting biotopes. Much less often they are used for placing nest Aster nigunda, Morus nigra, etc. Amorpha fructuosa and Copis form of trees die in the first year after placing nest of them. Sambucus nigra is most stable and, depending on the intensity of the impact, can vegetate for several seasons, but in the future it also dies. Water birds, such as cormorant, is very important links of some food chains and a factor that facilitates the dislocation of matter between aquatic and terrestrial ecosystems. From the one perspective, cormorants exclude biogenic elements along with fish from aquatic ecosystems. Also, cormorants hunt at the very large distance from colonies. Within a radius of up to 30 kilometers, they leave phases in a relatively small area below the colony. Nesting colonies of fish-eating birds affect the habitats and phytocenoses in different ways. During the breeding season, cormorants transfer a large amount of biomass and chemicals from the aquatic environment to colonies located on land. The consequence of the introduction of allochthonous matter into the terrestrial ecosystem can be significant enrichment of soil and plants with nitrogen, phosphorus, and potassium. In addition, cormorants break branches during nest construction, resulting in defoliation of the soil under trees. In areas with high biodensity, phases cover herbaceous vegetation, limiting photosynthesis and ultimately leading to plant extinction. The relationship of the great cormorant with other water birds depends, first of all, on the characteristics of the species that nest in mixed settlements with the great cormorant, as well as the specifics of the nesting sites, the type of nesting biotope, the lack of suitable nesting areas, etc. The impact can be both direct and indirect and can be both negative and in some cases positive. The analysis of available data on the 56 species associated with the great cormorant in 20 mixed settlements allows us to state that, depending on the colony, the composition of the avifauna complex can vary greatly, ranging from 3 to 20 species. On the oppositional speed, the growth of great cormorant population affects the species composition and the number of other settled birds. Since 1993, the cormorant has gradually displaced such species as Greta alba, Greta garzetta, and Ardea cinerea, as well as Sterna hirundo and Sterna albifrons from the nesting sites. In conditions of scarcity of nesting sites on the islands, only Larus cahinans can nest together with the great cormorant, with its high number. The intensive increase of the number of cormorants of the speed provides, in addition to Larus cahinans, a number of other species, in particular Pica pica and Corvus cornix, as a food base. Corvus cornix is also an active predator of cormorant eggs and young nestlings. This cooperation between Corvus cornix and Larus cahinans has often been observed. When gulls raid the colony, scaring the cormorants and forcing them to leave their nests, the nests are simultaneously visited by crows, which take out eggs and nestlings. The increase of the number of Corvus cornix on the Abitichna spit is directly correlated with the increase of number of cormorant colonies observed in recent years. We observe a different situation in Sivash. The competition of species for nesting territories determines the conditions of their existence, which we traced on the island of Central and Eastern Sivash from the great cormorant and Larus cahinans as the most massive colonial species. Research, retrospective analysis, and our data covers the period from the late 1990s to 2021, when the location, number, and species composition of colonial bird settlements were characterized by the method of absolute records. The main perennial nesting sites of this pair of species have been identified. In central Sivash, this is Kitai Island, and in the east, the islands of Chonhar, Koenli, Arabat Spit, Soleprom, and Polygonna Spit. Great cormorant is strong territorial competitor, and the places occupied by the colonies of this species become inaccessible for nesting of most of traditional for the island's colonial Haradriiformes species. In addition, great cormorant nesting sites attract many Larus cahinans to the island, whose predation usually makes nesting of the same island almost impossible for other species of gulls and terns, with the exception of Larus ichthyoetus and Sterna hirundo. The great cormorant has a definite positive effect on Pelecanus anacartalus. For this species, cormorant colonies are the marker of the security of the nesting territory, and it turns to be an important factor in the restoration and further preservation of nesting settlements of Pelecanus anacartalus in the Black Sea Biosphere Reserve. Another species on which the great cormorant has a positive effect on the islands of Tendra and Yahorlik Bays is Larus cahinans. At the first stages of the formation of nesting settlements, the great cormorant displaces the gulls from their traditional nesting biotopes in the lower part of the island, whereas the significant death of its nestlings due to the rushes was noted. Colonies of great cormorants are a very rich source of food for gulls, which they obtain both by kleptoparasitism, passive and active, and by predation on eggs and nestlings. An analysis of both published and original information concerning the great cormorant in the region allows us to draw the following conclusions. The activity of cormorants affects various components of the natural complexes of the Azov Black Sea region of Ukraine. Since the great cormorant is an obligate ichthyophag, it exerts a certain influence on the fish productivity of the coastal zones of the Azov and Black Seas. Fish resources experience a heavy load from the species, especially during the feeding of nestlings. Its impact on herbaceous vegetation is manifested both in a change in species composition of phytocyanosis and the subsequent disappearance of vegetation in certain areas. Tree vegetation is also negatively affected. Mechanical damage of trees by birds when collecting branches for nests, the destruction under the weight of nests, drying of trees due to the effect of large doses of nitrogen and phosphorus compounds from birds' excrements, and in the case of a large area and number of cormorant colonies, the drying of trees occurs very large areas which can reach tens hectares, which undoubtedly causes damage to the forestry. The high number of cormorants leads to a reduction in the number of some other species of birds. Improvement of nesting bird complexes, herons, terns. In the conditions of a shortage of nesting sites on the islands, only Larus cahinans can nest together with the great cormorant with its high number.
Okay. Okay. Thank you for the report. Dear colleagues, do you have any questions? Please.
Thank you very much. Can I ask, I would like to ask to Mr. Sidorenko. Andrei, are you with us?
I'm there.
Yeah, okay. Thank you very much for your presentation. And I would like to ask you about trends of population of great cormorant in the south part of Ukraine. What do you think about trends? Is increase, decrease, or stable population? I mean, only the Azov Black Sea coast.
I think now during 2021, trends of population of great cormorant in Azov Black Sea region of Ukraine is decreasing because we observe decreasing of number of colonies, number of birds in it, and I think that now there is only quarter, decreasing is to quarter of all population, but from 2022 we have no data about situation.
Yes, of course. Yes, of course. Thank you very much. And it's your registration this situation in all of local population,
At the most of population.
the most of population. Thank you very much. Thank you very much. And thank you very much, Mr. Rosacci.
Thanks. Any questions? Svetlana, please.
Okay, thank you for your presentation, and I have a small question. Have you ever investigated the colony after that great cormorant disappeared there? What happens with other species, with vegetation, if the great cormorant stopped to nesting there?
Thank you for the question. In some colonies, we observed that soil and herbaceous cover of the colony, even 2-3 years after great cormorant disappear, these plants and soil were in degraded situation. And not one second. Не быстро.
Okay, I understand.
Okay, thank you. Any questions?
No question.
Oh, thanks for the discussion. And next speaker, Maxim Kvitko, Nesterenko Alena Leholat, and Oleg Marenkov. And topic: accumulation of endocrine disrupting compounds in Procambarus virginilis tissue in Dnieper River, ecological and hygienic aspects. White minutes, please.
Dear conference participants, let me present a report to you, accumulation of endocrine disrupting compounds in Procambarus virginalis in Dnieper River, ecological and hygienic aspects. The author of project, Alena Lihalat, University of Custom and Finance, Dnipro, Ukraine, Oleg Marenkov, Олесь Гончар, Дніпро National University, Ukraine, Олесь Нестеренко, Олесь Гончар, Дніпро National University, Ukraine, Тетяна Лихалат, Олесь Гончар, Дніпро National University, Ukraine, Максим Квитков, Kryvyi Rih State Pedagogical University, Ukraine, Alexander Kabryushko, Kryvyi Rih State Pedagogical University, Ukraine, Yuri Lehalat, Олесь Гончар, Dnipro National University, Ukraine. Next slide, please. Ecosystem pollution is a serious problem worldwide. Aquatic ecosystems are extremely important for biota, fauna, flora, and humans. In recent decades, there has been a worldwide concern for the environment due to the difficulty of removing pollutions through traditional water and wastewater treatment processes. Among environmental toxicants, endocrine disrupting compounds encompass nearly 800 different chemicals, including both natural and synthetic compounds, including pesticide chemicals in consumer products and food contact material, pharmaceutic and personal hygiene products. Next slide, please. The marbled crayfish Procambarus virginalis belongs to freshwater crayfish species. Procambarus virginalis is a purple pet species in Europe and North America. Procambarus virginalis is an invasive species in the Dnipro River in Ukraine. Now the species has been introduced into the natural ecosystems of the Dnipro near to the city of Dnipro. Through the parthenogenetic methods of reproduction and polytrophic omnivorousness, Procambarus virginalis can withstand low water winter temperature in the European temperate zone. Next slide, please. Studies indicate the need for monitoring to assess certain classes of pollutions, such as BiodES, which are known to release serious toxicological hazards to aquatic biota at low concentrations. The purpose of this work was to determine the concentration of hormones, mainly estradiol and cortisol, in Procambarus virginalis of different age as hydrobion from the Dnipro River in the city of Dnipro. Next slide, please. The concentration of estradiol and cortisol was determined by standard methods using standard reagents according to the instruction for the reagent set. Estradiol level was determined in caver embryons, young crustacea, carcasses, samples of Procambarus virginalis, and the muscles of 1 month old crustacea, 5-month-old and year-old ones. Cortisol level was fixed in gills and muscles of different age crustaceans, raw embryons, and juveniles of marbled crayfish. The statistical analysis was performed using the program statistic of Windows. Next slide, please. It is known that reproductive processes in crustaceans are controlled by the endocrine system. These hormones play a significant role in normal growth and development. Water pollution can impair reproduction, development, immune response, and other physiological processes which can ultimately affect the survival of aquatic organisms. Next slide, please. Epigenetic programming established during development can be altered by exposure to ICSF during sensitive periods such as the preovulatory and ovulatory phases and cause immediate adverse outcomes. In addition, changes in methylation profiles and acetylation landscapes that occurs during these periods may predispose to the development of pathologies that occur later in childhood or adulthood, and more dramatically may ever be transmitted from the generation to generation. Next slide. According to the results of our research, the average level of estradiol in scale samples of Procambarus virginalis is shown on this slide. These results highlight the ubiquitous bioaccumulation of estrogens in aquatic invertebrates depending on the duration of exposure. Next slide, please. The distribution of estradiol in the muscles of Procambarus virginalis can directly and effectively reflect the effects of EDC. Xenoestrogens can act on several mechanisms simultaneously. Estrogenic xenobiotics also disrupt the physiology of aquatic organisms, biochemically processes as triggers of oxidative stress can affect their reproductive development, cause transcriptional effects, and there are also quite powerful initiators of signal cascades from membranes. Transfer of estradiol from the digestive system of Procambarus virginalis to muscles may pose a potential risk to aquaculture, wildlife, and human health associated with the consumption of aquatic meat. Finally, they contribute to the general accumulation of estrogens in the environment. Next slide, please. Crayfish gills play an important role in the transport of respiratory gases, extraction of nitrogen compounds, and osmoregulation. The gills are the first barrier to waterborne pollutions. Gills' tissue condition is generally considered a good indicator of water quality and suitable for environmental impact assessment. Its changes can lead to a decrease in oxygen consumption and a weakening of the function of osmoregulation. A marked decrease in oxygen consumption can lead to internal hypoxia, which affects metabolism and movement. Next slide, please. Based on the results of study, it was established that the level of cortisol in the gills of Procambarus virginalis increased depending on age. It's shown to the figure 1a and 1b. In this figure, we can see the content of cortisol in teaser, gills, muscles, roe, embryons, and juvenile of marble crayfish. Significant difference between the cortisol content in roe, teaser of embryons and juvenile of the investigated crustacés wasn't found. It can be assumed the hormone level changes in this sample are related to the direct influence of the surrounding water environment. Next slide, please. The obtained results emphasize the widespread bioaccumulation of BiodES, namely compounds with estrogenic activity in aquatic invertebrates, depending on the duration of exposure, which may indicate the role of the aquatic environment in the bioaccumulation of xenoestrogens. The study demonstrated the need to monitor exposure to EDC contaminants due to the effects on water quality, degradation, and serious toxicological effects on aquatic biota and human health to inhibit such potential. Estrogens are clearly necessary for normal fauna and human physiology, but can have serious negative consequences if allowed to accumulate in the environment. Thus, it is possible to note two important aspects related to the issue of contamination of aquatic biota with compounds that cause the destruction of the endocrine system. The first aspect concerns the impact of BiodES on the reproduction of wildlife populations, including aquatic organisms, which may have a negative impact on species conservation. The second aspect is that since the effects of EDC are realized on humans in different ways, primarily through trophic link food chains. Further research is needed for public health policy planning. Thank you for your attention. Please send you.
Thank you. Questions, please. Any question? Okay, please write questions to text chat. And next speaker Svetlana Vinokurova, and topic biodiversity assessment of the Danube region as a tool for the development of protected areas in the region. 5 minutes, please.
Dear colleagues, let me introduce the result of the investigation of the group of researchers, which names you can see on the screen. The main idea of this research is the using of the geographical information approach to the assessment of biodiversity. Until recently, the most common approach to an assessment of biodiversity was the using various indexes, like Shannon, Simpson, Margolef, etc. But geographical approach has much expanded the practical application of the data. Nowadays, the methods of geoinformation systems for assessment of biodiversity are widely applied all over the world. We try to do this assessment in the Danube region of Ukraine and consider the possibilities that this kind of assessment provide for nature conservation. Danube region, one of the most valuable area. Thus, the Danube Delta was included by the WWF in the list of 200 most valuable and endangered ecoregions of the world. So it's no surprise that most of researchers concentrate the investigations within the boundaries of the Danube Delta. We consider here the Danube region in a broad aspect, based on history of the development of the delta, taking into account the past natural connection of the small steppe rivers and the Danube lakes with the unique Danube delta. So we have a comprehensive view of the region. And from this point of view, the region consists of the Danube delta and adjacent small river basins, with different landscape types that contribute to the development of rich biodiversity. The area also includes the Danube lakes, which used to be estuaries, Kagul, Kartal, Kugurluy, Yelpuk, Katlabuh, Kitai, and adjacent areas of Lower Danube. Vertebrate taxa, such as fish, reptiles, amphibians, birds, and mammals, except for Chiroptera, were chosen as biodiversity indicators. Compared with other taxa, they are more often on top of the ecological pyramid and are sensitive to the slightest changes in ecosystems. Using QGIS methods, a regular grid net was made for the region, consisting of 200 hexagons, each with an area of about 5,000 hectares. A gradient scale was developed to assess species diversity of each taxa. Given the significant differences in species abundance among selected taxas, ranked scores were standardized. Therefore, the data from zoological studies were correlated with the regular net of hexagons. For the integral assessment, the method of hotspot was also used. It should be noted that the region has suffered from considerable anthropogenic transformation. West coastal areas were embanked and converted into agricultural lands. Small rivers were transformed into a set of reservoirs, ponds, and canals. Most of these activities had a negative impact on the integrity of natural communities and led to decrease in regional species abundance. The status of selected taxas of fauna makes it easy to judge the environmental process in the region, and the concentration of their species abundance can diagnose the degree of importance or transformation of various spatial areas. The analysis of the region transformation used the open data on land use classification posted by the European Space Agency. Let's consider the obtained results. Fish. In most drainage basins, the fish species composition is extremely poor. Small rivers are drying up, but the embanked areas still preserve some species abundance and their middle reaches. Only in Kagilnik and Sarata rivers, the eastern ones in the figure, the water content of which is higher, species diversity persists throughout the entire length of their channels, but it's relatively low. Amphibians and reptiles. Suitable habitats for reptiles and amphibians are located not along the slope landscapes of small rivers, but also along numerous forest belts in agricultural lands. Birds. Birds is the basis of the species riches of vertebrates in the region. The avian diversity is represented by more than 320 species. Due to their plasticity and mobility, birds can quickly disperse within the Danube region and outside its boundaries. Mammals. The area of high mammal diversity are concentrated in latitudinal zones along the Danube floodplain and in the north of the region. This diversity is most affected by the heterogeneity of landscape and habitats. Thus, the northern part of the Danube region is located on the elevation, streaked with channels of small rivers, gullies and ravines, with Tarutinska steppe and forest areas. This determines the high species diversity of mammals, mainly rodents. The landscape of the Danube floodplain are highly mosaic, with large areas of riverine forests, meadow ecosystems, and floodplain lakes. That ensures a high species abundance of all order of mammals, typical for the region. Moreover, the border control and several reservoirs are located here, contribute to the conservation of biodiversity. An integrated assessment of the whole zoological diversity illustrates the cohesion of this natural region. The absence of any hiatuses and clear latitudinal demarcation lines from the Danube floodplain to the small steppe river basins. However, some biodiversity loss is recorded in the interfluvial areas and the middle reaches of the rivers flowing into Lake Kotlabukh and Stansovsko-Zhebrianovsky Plavny. The smallest percentage of naturalness is noted also for this region, for the rivers of Kotlabukh Basin and the Stansovsko-Zhebrianovsky Plavny. This probably explains the least biodiversity in this area for the whole region. Further steps of biodiversity conservation. Geospatial assessment of the ichthyofauna clearly shows the consequences of water flow regulation in small rivers. The current negative situation can only be solved if the water exchange between the small rivers and the Danube floodplain is restored. Natural state of channels and floodplains of small rivers support the survival of many animal species, including Ukrainian red-listed ones. Exactly small rivers and their floodplains enabling the potential expansion of the genetic exchange corridors between fragmented populations and subpopulation of animals. So the restoration here is strongly recommended. The critical destruction of steppe and meadow areas converted to agricultural lands has led to the loss of many stenobite animal species. That means that the other step of biodiversity conservation is the restoration of steppe and meadow areas. The obtained data of the biodiversity distribution enable the identification of gaps and the development of a more effective system of protected areas. The data analysis indicates the need to create at least 3 new reserves and to expand the regional ecological network. Conclusion. A large-scale geospatial assessment of the Danube region biodiversity has allowed identifying the most valuable areas, naturally located in the Danube Delta and the lower part of the Danube lakes, and also the areas with an urgent need of restoration, which we discussed earlier. A holistic view of the region also showed the role of small steppe rivers in the species exchange and the formation of ecological corridors. Despite the significant transformation, the Danube region still remains an important area for biodiversity, not only its key part, the Danube Delta, but also within the boundaries of adjacent small rivers basins. The use of a holistic spatial approach to biodiversity assessment provides wide opportunities for management decision-making, such as identifying potential protected areas, areas for restorations, etc. This research was carried out within the framework of the European Endangered Landscape Programme and the project Renewal of Wetlands and Steppes in the Danube Delta region, which is being implemented by Rewilding Europe. The authors are grateful also to Dr. Joseph Chernichka for his valuable advice in preparing the manuscript and presentation. And Thank you very much for your attention.
Thank you. Dear colleagues, do you have any questions? Please.
Thank you very much, Svetlana, for your presentation. I have asked you about one moment, okay? Okay. To my opinion, the assessment of biodiversity is an impossible goal, because it's very difficult to study, because it's different type of study, because it's different complexes of biodiversity. But your presentation showed to us everything is possible. Thank you very much. But I have a question to you about compare of biodiversity of Danube Delta with another delta estuary of big river in south part of Ukraine, for example, Dnieper River. Do you have, I know you have not database material for example, Dnieper, but I ask you about expert of this assessment. And do you have compared Danube Delta with Dnieper Delta?
This information vegetation here. Is my problem?
Yes, I hear you.
Okay. Compare the Danube Delta. This kind of assessment for them to this kind of assessment knowledge level. It's interesting, really interesting, and about this comparison. Critically important to take for one so we need to more data. Question but it's difficult and careful to take a lot of information from this campaign. And we don't do
okay, thank you very much. And another not question, just proposition for your team, for team of author this presentation. In your study, not include bats like mammals. We have material for bat activity during the several seasons, and for example, spring season and autumn season. If you need, to continue a study in this type of assessment, we can send you our material is the base with, I don't know, more than 5,000 records in database only for bat activities in Danube Delta. If it's interesting, we can send you.
Thank you. Yes,
Thank you. Any questions? Thank you for our discussions. And next speaker, Ольга Пюрко, Тетяна Христова, Владислав Пюрко, Людмила Арабаджи-Типенко from Bodin Khmelnytskyi Metropolitan Pedagogical University, and topics see similarity of culture and nature flora under chloride lowered in conditions of southern Ukraine. White, please.
Good afternoon, dear colleagues. Let me introduce to your attention the results of Ukrainian science group on the seed similarity of cultural and natural flora underchloride load in conditions of southern Ukraine. The structure of the presentation includes the following components: introduction, aim, methodology, results, conclusions. Due to increasing anthropogenic pressure on the environment, the climatic conditions on the planet Earth are changing. Saline soils make up almost 25% of the Earth's surface. Unfortunately, the areas tend to expand due to the climate aridization and increase in the average annual temperature of the planet. The south of Ukraine forms a risky agricultural zone which is characterized by a temperate continental climate with hot summers, high solar insolation and significant water deficit. Seeds are an equal product of evolution, the main generative organ of plants, characterized by a number of morphological, physiological and biological features, among which there is adaptation heterogeneity. Therefore, research on the similarity of plant seeds to salt stress is becoming increasingly relevant. Thus, the research aim is to find out the seed similarity of natural and cultural flora under chloride load in southern Ukraine. During the theoretical study of the problem, research and experimental work, general scientific research methods were used. Theoretical analysis and generalization of scientific and methodological literature on morphology, ecology, and the functional peculiarities of plant seeds and periodicals, vegetative and experimental methods, generally accepted physiological methods, quantitative and morphological analysis, mathematical and statistical processing of the obtained data. The choice of the chloride salts concentration is explained by the fact that such a percent is considered stressful for the seeds, taking into account the physiological, biochemical, and anatomical morphological features. The representative selection of cultivated and wild flora was determined by the prevalence of species, taking into account the soils and climatic conditions of southern Ukraine. The obtained experimental data showed that the seeds of cereal cereals sprouted 93% in aqueous solution for 3 days, 89% in 0.1% natri chlor solution, 95% in 0.2% natri chlor solution. The researchers' data presented that the seeds of Triticum durum sprouted well in natri chlor solutions with a concentration of 0.1% and 0.2% by the third day. Seed germination of Avena sativa showed that on the third day the number of sprouted seeds in water was 50%. On the fifth and seventh day it increased to 99%. The obtained results proved that the germination of Hordeum vulgare seeds began on the third day, with the highest values observed in 0.1% natri chlor solution is 95%. In the aquatic environment, 93%. The faster the germination process occurred with the seeds that germinated in water, the longer the germination occurred in solutions. Our studies showed that during the germination of beta vulgaris seeds, the first sprout appeared on the fifth day in the pure water, up to 30%, in 0.1% natri chlor solution, up to 20%, and the smallest amount was observed in 0.2% natri chlor solution, up to the 2%. Therefore, in beta vulgaris, the best germination occurs in water, and in 0.2% natri solution the worst. It is found that the seedling of Phaseolus vulgaris began to appear on the third day in the sample with the water and in 0.1% natri chlor solution. On the fifth and seventh days, in all samples, the number of sprouted seeds increased. According to the results of seed germination of Helianthus annus, it was found that for 3 days the most seeds germinated in aqueous medium. In 0.1% natri chlor solution is 60%, and 0.2% natri chlor solution is more than 20%. On the 5th and 7th days, 100% of the seeds sprouted in all samples. It is proved that the rate germination of Helianthus annus seeds is characterized by inversely proportional dependence on the salt concentration in the medium. Seed germination of Valeriana officinalis was observed for 12 days, but during this period the highest values were obtained in the aqueous environment, the lowest in 0.1% and 0.2% saline solutions. For the anti-germination period of Matricaria recuta seeds, which was 7 days, the highest values were observed in water. The average values were in 0.2% natri chlor solution, and the lowest values were in 0.1% natri chlor solution. The researches of the salt grammaceous seeds of natural and cultivated flora in the conditions of southern Ukraine showed that the number of germination days, the environment, and the definition of sodium and chlor ions influence determined the opportunity distribution of the studied plants. It has been proven that the diagnostic signs of florid grammaceous were most clearly manifested in representatives of cultivated flora than in natural ones, which is due to the realization of the relationship between the genetic program and phenotypic manifestations that are associated with the special climatic conditions of southern Ukraine, which is of practical importance when developing strategies for cultivation, agricultural and decorative crops on saline soils, and in the future should be taken into account when selecting a plant assortment for the land of southern Ukraine. Thank you for attention.
Thank you. Questions, please. No questions. Very bad. Very interesting report and many scientists in our workshop. Because please write the questions to text chat, and we go next report. Information provision for monitoring the sustainable development of the land and biodiversity. Автор стаття начала Олексій Карипанов, Лілія Лазебнюк, Дар'яна Черненко, Георгій Карипанов. Speaker Юлія Лазебнюк. Why, please, I apply presentation.
Good day, everyone, and welcome to our presentation. First of all, I want to thank you all for coming here today. And now let me present to you the results of our research of information provision of monitoring the sustainable development of the land and biodiversity. During this presentation, we'll consider the main problems of monitoring the sustainable development of the land and biodiversity, the main databases we can use for this, and the main approaches to appropriate data collection. In this report, we would like to concentrate on the fact that there are completely different approaches to data collection and construction of biodiversity observing system in the world and in Ukraine. First of all, it must be said that biodiversity is a key theme of the United Nations Agenda for Sustainable Development 2030. Our research is primarily related to monitoring the achievement of the Sustainable Development Goal 15, abbreviated SDG. In order to achieve this goal, conceptual block diagram of information provision for the regulation of sustainable development of ecosystems and biodiversity was shown in our research. It characterizes structure and sequence of operations aimed at justifying management decisions. It should be noted that the main phase of this diagram is formation of a unified system of metadata that are international standards, classifications, and unified definitions of phenomena and processes that summarize modern world experience. A big problem is that many international statistical classifications are not used in Ukraine. During the research, many differences were found between indicators for micro, meso, and macro levels. Such differences make each set of indicators interesting to study. A necessary condition for ensuring a systematic study of sustainable and responsible use of ecosystems and restoration of biodiversity is the formation of a unified open database as an information source for ensuring management. This database should be able to be used at micro, meso, and macro levels. And now I want to tell you about microdata. Company reporting is an important source of data for monitoring system. Accounting data for the formation of key indicators should be collected at the level of an institutional unit and aggregated by the region. Institutional units usually consist of several different units that produce different products or services and have different locations. In the system of national accounts, they are called establishments or local kind of activity units, LKAU. This is especially true for large enterprises. It is useful to collect and compile environmental indicators for each facility first, and then aggregate the reporting of institutional unit. It was about approach to data collection. And now let's see what about the indicators. Development of a unified list of main indicators for reporting by institutional units was initiated by United Nations Conference on Trade and Development in 2016 and extending it in 2017, 2018, and 2022. In 2022, in the ecology group of indicators of the micro level, was added subgroup B6, land and biodiversity, which includes indicator B6.1, lands used near biodiversity sensitive areas. This indicator is defined as you can see in the slide. Among the potential sources of information, the following were identified. The first, information about the location of key biodiversity areas can be found on the official site of KBA partnership by the link on the slide. It should be noted that for Ukraine, the information is presented for the year 200. Second, information on protected areas can be found by searching the Protected Planet Global database. You can see the link on the slide. Let's now look at the next slide. Special attention should be paid to the fact that now it's impossible to receive reliable results of biodiversity research without joining effort of biologists and modern specialists in statistics. The data revolution, which includes the open data movement, new ICTs for data collection and the availability of big data, together with the emergence of artificial intelligence, has led to a radical transformation of society. Real-time mobile population positioning data and high-frequency satellite data are just main examples of big data that can be used to monitor the achievement of SDG 15. This leads directly to our next point. One of the main projects aimed at integrating the development of big data for monitoring, evaluation, and policy development within Group on Earth Observations Biodiversity Observation Network is United Nations Biodiversity Lab, a geospatial information platform that maps natural sources and monitors environmental risks in real time using existing data and new digital technologies. As part of the above-mentioned project in 2022, the calculated data sets and indicators were included in the existing decision support system based on the United Nations Biodiversity Lab. Project data and indicators will be regularly updated until 2030 through the Biodiversity Lab. The results of the project are expected to provide a modern digital foundation for scaling up this forecasting methodology to more countries around the world, working to monitor progress towards SDG 15. The project envisages a widespread use of data sets in 170 countries. That is presented on this slide. On the next slide, for example, the results for several main blocks are presented. Slide 10 shows changes in the forest cover in Ukraine for 2001 to 2020. Many organizations deal with forest change issues, so the definitions related to this concept are important and should be unified during the information provision of the study. Also, slide 10 shows Ukraine's land cover in 2020. The European Space Agency and Copernicus Climate Change produced global land cover maps from 1992 to 2020. The map legend contains 22 land cover classes that are briefly defined using the land cover classification system developed by the FAO. Biodiversity Intactness Index reflects the results of modeling the average number of originally present biological species expected as a percentage of their total number in a pristine ecosystem. Initially, the data was only available for 2015, but is now available in a time series covering the period 2000-2015. To summarize everything we mentioned, I want to say that information provision for monitoring the sustainable development of the land and biodiversity should be based on an understanding of the integrated use of statistical administrative data sources, metadata for their qualitative use, as well as a combination with big data, such as satellite images. The algorithms of data processing, modeling, and construction of integrated indicators would be the subject of our future research. Thank you for your attention.
Thank you. Questions, please? Any questions? Okay, I have questions. Yulia, are you ready?
Yes.
I have questions, 2 questions. First questions, explain in more detail about problems of the use of land-related classification in Ukraine. And two questions, which data sets of indicators are essential for biodiversity monitoring in Ukraine and why? Thank you.
Thank you for your question. That's really a very important problem, especially for Ukraine. In the world practice, land classifications are usually developed in two directions, land cover classification and land use classification. However, in our research, it was found that in Ukraine, the specified groups of classifications are not used now. And they have been replaced by categories of land according to their intended purpose, in accordance with the land code of Ukraine. We have really a great need to implement these two classifications in Ukraine and to use them for gathering and analyzing the data. About the second question, about indicators that are essential for biodiversity. In my opinion, the biodiversity intactness index is essential for biodiversity monitoring in Ukraine. This index is an estimated percentage of the original number of species that remain and the abundance in any given area, despite human impacts. The index uses the most comprehensive evidence base of its kind alongside robust peer-reviewed methodology. Very important is to note that biodiversity can only be managed if properly measured and modeled. Thank you.
Thank you. I propose a small change to our program, and the next speaker, Victor and Natalia Demchenko, and topic: a system of environmental important decision making for the sustainable use of marine estuaries in the conditions of anthropogenic and climate changes. Wait, please. I play demonstration. Okay.
Dear colleagues, the United Nations has declared years between 2021 and 2030 as the International Decade on Ecosystem Restoration. The goal of this decade is to bring together all interested parties to prevent, halt, and reverse the degradation of ecosystem on every continent in every ocean. This initiative provides many areas of work that will draw attention to the problem of restoration ecosystem and ensure their sustainable use in the future. The length of Azov-Black Sea coastline in Ukraine is about 1,500 kilometers, which is just about 25 large limans of the northwestern Black Sea and Azov region. A huge part of coastal marine ecosystem and limans falls under the natural protection territories of the Azov-Black Sea region. Is given the status of wetlands of international importance, and it's considered an element of the Emerald network, although they are important from the social and economic sides as well. Besides, these waters was recreational value. More than 15 million people came to the coast for vacation each year. As a result of water and marine framework standards implementation in Ukraine, it became possible to assess the current ecological state of coastal water bodies and largest limans in the ESC categories, based on morphological and functional indicators of benthic vegetation. Due to the analysis of ESC, was obtained for most of the region's water areas, which makes it possible to identify the most problematic water bodies that primarily require complex management to achieve GAS. For a long time, the management of these water bodies took place spontaneously and was aimed at the transformation of various ecological states. Most of the feasible anthropogenic transformation have changed the course of natural process in these ecosystems. Some ecosystems were radically transformed, isolated from the sea waters, changed both at the level of abiotic characteristics and at the biocenosis level. This is why the purpose of a systematic approach to the environmental management process, the main element of which should be a multi-component decision support system. This scheme will enable to develop clear plans for the restoration and sustainable use of marine ecosystem, as well as to prevent negative changes and degradation of marine water areas. Despite the obvious need to develop a systematic approach to design support in estuary management, there are currently no complete step-by-step schemes that allow for a comprehensive solution to the task of ecological management. The main defects and problem of modern ecological solution implemented in Ukraine are: first, lack of systematic scientific studies on the state of water bodies, which should be implemented on the basis of the basin approach and the requirements of the Water Framework Directive. Second, lack of works on the assessment of ecosystem services as a tool for understanding the ecological, social, and resource value of water areas. Third, insufficient use of elements of the mathematical modeling of hydroecological and socio-economical processes. And fourth, lack of understanding of the need to involve experts of appropriate qualification by the customers and their unwillingness to finance the full scope of the development and the design stage. Given the lack of systemic solution in the management of liman and coastal marine waters, there is a need to develop a holistic step-by-step scheme that will comprehensively address ecological management using a basin approach, ecosystem approach, and ecosystem services. The basin approach assumes that the catchment area, together with the reservoir, forms a holistic geo-ecosystem with transit accumulative functions and directly affects the formation of ESC hydroecosystem. According to the World Water Framework Directive, the river basin is the main territorial unit when it comes to protection of the aquatic environment. Planning and sustainable landscape management structure includes special zoning taking into account the morphology of the terrain and the spatial location of lands among themselves. Formation of a sustainable ecological frameworks along with an ecological network consisting an environment stabilizing and refuging lands. Creation in an environment protected regime. Ecosystem approach. The main idea of the ecosystem approach lies in the fact that the natural anthropogenic components are considered to be interrelated elements in a holistic system. At the same time, the main goal of ecological management is to maintain the structure of functioning of the natural system, while the ecosystem itself together provides its services as a social benefits that are essential to society. In fact, successful ecosystem approach amplification is centered around the concept of health by achieving both the health of nature system and the health of human system. Ecosystem services as a tool for the basic diagnosis stage of coastal marine waters and liman's aims to assess the value of aquatic waters and to provide their ecological, social, and economical features. Among many ecosystem services are those that provide resources: food, water, timber, raw material, regulate processes in ecosystem, climate impact, flood and natural disaster control, water quality, and so on. Provide cultural and social services, for example, aesthetic and spiritual value of nature, and supports ecosystem functioning, soil formation, photosynthesis, nitrogen cycle, and so on. At present, there are different methodological approaches for assessing ecosystem services. The Ramsar Committee, as part of wetland management, proposed a rapid assessment of ecosystem services. The proposed scheme has 3 blocks: a data collection block, solution research block, and the measure implementation block. Those blocks are sequential and must be implemented in stages. Data collection blocks involves an implementation of several stages. First, assessment of the current state of the water body. Second, definition of the problem and their ranking. Third, evaluation of the ecosystem services. The second block of the systemic decision-making scheme for estuary management is based on the implementation of the decision support system. The essence of the management process is to make a decision. Is it the choice of a course of action from a set of existing alternative options? In such conditions, the problem of choosing the most rational and effective solution, is it the best solution, became one of the most important tasks of production management. The third block is a systemic decision-making scheme for estuary management, is dedicated to implementation of four stages. The first, environmental assessment of decision and projects. Second, implementation of environmental measures. Third, efficiently evaluation. And the fourth, research for new solutions and their improvement. As a conclusion, the Azov Black Sea coast is presented by the significant number of estuaries and bays, which function for a long time thanks to artificial connection with the sea. Most of them are included in the nature protection territories of Ukraine. On the other hand, they are important from the social-economic point of view. For a long time, the management of these water bodies took place spontaneously and was aimed at the formation of various ecological stages. The lack of the systemic concept of the development of estuary management plans does not make it possible to make the necessary management decision. Developed decision-making scheme for estuary management is based on the implementation of successive stages, which is a multi-component decision support system functioning in its center. This scheme is cyclical and requires contained improvement of the decision made and evaluation of the effectiveness of measures. Thank you for your attention.
Thank you. Questions, please? No questions? I have questions. Okay. First questions. What are the prospects for implementing the approaches in Ukraine? And 2 questions, please. Is there an effective methodology for evaluating ecosystem services? Natalia or Victor?
Victor, thank you for questions. 4 or 5 currently, estuaries and not management in Ukraine, chaotic action lead to degradation of the water bodies. it is necessary to develop normative acts for the implementation of ecological management of estuaries. Ecosystem service in Ukraine must methods have generation approaches and will not allow quantity assessment of the rule of each ecosystem service. Thank you.
Thank you. My questions, please. Okay. Next speaker, Natalia Suryadna, and topics chromosomes and Pelophylax ridibundus pallas 7071 in different population systems of southern Ukraine. Why, please, I play presentation.
Dear conference participants, I present to your attention the results of the study of chromosomes of Pelophylax ridibundus in different population systems of southern Ukraine. Southern Ukraine is characterized by unique biological and genetic diversity. It is home to common and rare species. They retain their relict nature and have special evolutionary mechanism. The hybridogenetic speciation is of particular interest here. This requires a new strategy for biodiversity assessment, conservation, and reproduction. Pelophylax ridibundus is the most widespread type species of amphibian in bodies of freshwater of southern Ukraine. It is one of the parent species of the hybridogenetic complex of green frogs of the genus Pelophylax. The vast majority of pure ridibundus populations are distributed in the southern and southeastern parts of Ukraine. In orphan regions, Pelophylax ridibundus occurs in combination with Pelophylax lesone and the hybrid taxon Pelophylax esculentus. You can see of the maps the distribution of different population systems of green frogs in Ukraine. On the first map, mixed population. On the second map, pure populations. The karyotype ensures the stability of the species. It is important to investigate how karyologically stable this species is in the conditions of the southern Ukraine. There are investable genetics mechanisms that may have great potential for understanding the role of hybridization as a cause of new species. The purpose was to identify specific characteristics at the level of karyotypes and to conduct a comparative morphological analysis of Pelophylax ridibundus chromosomes from different populations of South Ukraine, include the Crimea. 8 individuals of Pelophylax ridibundus were used in the study. For comparison of karyotypes, typical populations of Pelophylax ridibundus were selected. Point 1. Kherson region genetics Genический district Arabatspit. Point 2. Kharkiv region Izum district near Snishkivka village. Point 3, Kherson region, Golopristan district near Starosbulivka village. And point 4, Crimea, Bakhchisarai district, Zalisny village. A total of 49 metaphase plates were analyzed, 20 of which were selected for morphological analysis. The standard karyotype of Pelophylax redibundus is symmetrical and represented by 2 end chromosomes. The karyotype include 5 pairs large and 8 pairs of small chromosomes. The analysis revealed certain variability and heteromorphism of individual homologs and chromosome pairs. The chromosomes of redibundus type population systems are relatively more stable in the quantitative and qualitative terms. More variable chromosomes in the karyotypes of Pelophylax redibundus of mixed population systems, Redibundus esculentus and Redibundus esculentus lysonum. The slide shows the normal karyotypes of the studied populations. This can be represented by chromosomes of different sizes but the same morphology. Heteromorphism exists among a group of small chromosomes. Incomplete sets are often found, which is typical for all analyzed populations. Homologs or even entire pairs are more often lost among small chromosomes. Among large chromosomes, homologs of the 5 pair may occasionally be lost. The chromosomes of redibundus type population systems are relatively more stable. A high degree of chromosome variability is found in mixed population systems. Here you can see the disturbed karyotypes, for example, no homologs on pairs 10 and 12 chromosomes. The results of the study suggest then the relative stability of chromosomes of southern Ukraine, Pelophylax redibundum in pure redibundus systems indicate a high potential for their resistance to both environmental factors and interspecific genetics balance. This is especially important in terms of preserving and increased genetics biological diversity, which mainly ensures the sustainability of ecosystem function. Thank you for your attention.
Thank you. Questions, please. No questions. Okay, I have a questions for the speaker. 2 questions. First questions, what factors can influence chromosome variability in amphibians? And second questions, please, what protection measures could be introduced for your research objects in southern Ukraine after the occupation? Thank you.
Thank you for the questions. About first questions. This can be external and internal factors. The variability can be influenced by genetic process during reproduction and the transfer of genetics material. This is especially relevant for green frogs, where, for example, the like frog is a kind of doctor of genetic material for the hybrid of a double frog with which it lives together. Environmental pollution, especially of water bodies, can also affect variability. But the like frog is the most resistant to such impacts. About second questions, the first measure should be to assess the state of the populations. The data we obtain will allow us to develop effective measures to restore biodiversity. Conservation institutions will be directly involved in this as they have all the necessary instruments for this. While the main problem we will face is meaning. Thank you.
That's a lot. Thank you. Any questions? Okay, let's move on to the next speakers. Иван Митаев, Виталий Хомич, Elena Дегтяренко, Петрова Шевченко, and Alexandra Markusheva. And topic: impact of mini hydropower on the chemical composition of water and phytoplankton of the reservoirs of the forest tip of Ukraine. Wait, please.
Afternoon, dear colleagues. Here is a report on the topic impact of mini hydropower of the chemical composition of water and phytoplankton on the reservation of the forest steppe of Ukraine. At the beginning and in the middle of the 20th century, small hydropower plants, MHPPs, were built on a large number of small and medium-sized rivers in Ukraine. In the 1930s to 1980s, there were estimated more than 1,500 of them. Now there are only 98 ones. The building of the reservoirs has caused radical changes in the ecological condition of rivers, due to the plans for significant expansion on hydraulic engineering on small rivers on the forest steppe of Ukraine. It is extremely important to find out the nature of the impact of MHPP on hydrochemical and hydrobiological regimes. It appears extremely necessary to develop measures to minimize the harmful effect of HPPs for optimizing the state of the reservoir ecosystem. This study is relevant in the framework of the European Framework Directive, AFD. The study was conducted at 7 MHPP reservoirs located on Koropetske River, Koropetske reservoirs, Nilый Tikach River, Zvenigorodsky and Natashevsky reservoirs, Kherský Tikach River, Yurpilsky and Mordashivsky reservoirs, and Rose River, Stabliyske and Korsun-Shevchenkivsky reservoirs. Slide 2. The selection of material was carried out during expeditions in 2013-2020, in the warm period from April to September. Determination of the species and quantitative composition of Aglaya was carried out jointly with the staff of the Institute of the Hydrobiology of the National Academy of Science of Ukraine, according to standard methods. Ecological assessment of surface water quality was carried out using generally accepted methods. According to the criteria of pollution by components of the salt composition, water in the reservoir on average was determined as having an index is 2.0, which applies to class 2 of the category 2, good clean. According to the ecological and sanitary indices, the average value of the water quality category of reservoirs was determined as 3.66 4.0, which applies to the class 4 of the category 4, satisfactory slightly pollution. Considering the biogenic substances, there was detected a significant excess of conservation of nitrates and phosphates. The MPC was observed for the following incidences. In the reservoirs Koropetske, nitrate by 3.5 times, Stebliske nitrate by 5.0 times, and Korsun-Shevchenski nitrate by 20.0 times, and ammonium nitrogen by 1.4 times. All this indicates significant decay processes of significant industrial and domestic effluents. Excess MPCs for phosphates are also related to this. The worst situation is observed in Stebliske, phosphates by 10.6 times, and Korsun-Shevchenkivske, phosphates by 10.2 times. Reservoirs. The high level by 3.4 to 8.6 times of phosphates were also detected in the rest of the reservoirs. Table 2. The above-mentioned hydrochemical environments play an important role in the formation of groups of aquatic organisms, primarily phytoplankton. The phytoplankton excessive development, eutrophication, correlated with a significant excess of the MPC for biogenic substances, especially phosphates, was observed. On the other hand, an ejection of the amount of phosphates leads to inhibition of the activity of algae, especially diatoms. As shown above, the amount of phosphates depends on a number of reasons. Among the most important are the size of the reservoir, the density of sediments out on the bank, and the area of reservoirs along with the flow. The same factors are observed highly influencing the phytoplankton abundance. The amount of phosphates is not significant in the small Koropetske reservoir, but the phyto predominates there. The quantitative ratio of different taxonomic groups apply in general and xanthophytes in particular defects in the reservoirs of the Kryvyi River. There is observed another pattern. Thus, one of the studies reservoirs on the Kryvyi River, Zvenigrodske, one is located within the settlements, and Lutashevsky reservoirs is located in sparsely populated areas. In addition, the Lutashevsky reservoirs is upstream, and Zvenigrodske one is in the middle part. Having almost equal amounts of phosphates in the reservoirs, Zvenigrodske reservoirs has 372 plus minus 23,000 cells per cubic decimeter of cyanophytes, while Lutashevsky reservoirs show the minimum number of cyanophytes, and up to their total absent in the upper and middle parts. Slide 6. Similar results were obtained on Yerskity River. Yurpilsky Reservoir is located in the upper reaches in sparsely populated areas. There is a minimal amount of biogenical substances and no cyanophytes. The Hodashevsky Reservoir is surrounded on two sides by settlements. Exceeding the MPC of phosphates was observed by 4 times. The cyanophytes were observed through the reservoirs. The number of diatoms was observed by 6 to 9 times lower comparing the previous reservoirs, size 7. The Stablivsky and Korsun-Shevchenkivsky reservoirs are located in the densely populated lower part of the Rose River. As shown above, there was observed a significant exceedance of the MPC for biogenic substances in these reservoirs. This significantly affects the species composition and abundance of phytoplankton, the number of cyanophytes and diatoms shown on slide 8. The relation between the conversation of nitrates and phosphates and the amount of cyanophytes according to the result of correlation analysis is characterizes are close. R is 0.868. The observed reservoirs were classified as beta-mesotropic and beta-mesotropic. Is in terms of cell probity as mesotropic, predominant type. In terms of tropicity, correlation between the density of cyanophyte groups and the amount of biogenic substances in the studies reservoirs on slide 9. Conclusions. In the term of the content of basic chemicals, the water of the reservoirs of the fourth step MHPP meets the state's fishery standards MPC in most indices. A slight increase of the MPC for magnesium by 1.09 to 1.15 times was observed in Yurpilsky and Korsun-Shevchenkivsky reservoirs. According to the criteria of pollution by components of a salt composition, water in reservoirs, on average, was determined as having an index is 2.0, which applies to class 2 of category 2, good clean. A close relation has been figured out between the concentration of cyanobacteria and the amount of biogenic substances in the water, which depends on the territorial location of the reservoirs. The effect of cyanobacteria on certain groups of algae and the other aquatic organisms has been elucidated. Thanks for your attention.
Thank you. Questions, please. No questions. I have questions for the speaker. One question. How do cyanobacteria affect other aquatic organisms? And 2 questions. What is the impact of hydroelectric dams on the hydroecological state of rivers?
Let me please answer to this question as a co-author. Thank you for your questions. For the first question, cyanobacteria consume a lot of oxygen, which changes some important water quality characteristics and reduces the concentration of oxygen which available to hydrobionts. In addition, cyanobacteria produce some toxic substances that are dangerous for many aquatic organisms, including different species of plants and animals. And as a result, environmental conditions of these organisms get worse. And for the second question, as a result of the installation of hydrotechnic buildings, the river step by step begins to transform into a lake. The water current slows down and the quantity of bottom sediments increases, including various harmful substances which accumulate in there. And also we can observe an intensive growth of cyanobacteria. Thank you.
Thank you. Any questions? Okay. Next speaker, Petro and Anastasia Gorlova. And the topics factor analysis of the wind farm, a wind park Donetsk, Ukraine, impact on the bus based on the index of their activity and dynamics of species diversity. 1 second, please.
Good afternoon, dear participants. Let me introduce you the presentation on the paper where we performed the factor analysis of the impact of a wind power plant on bats. This research was based on the index of their activity and dynamics of species diversity. The organization and implementation of monitoring of bats' existence conditions in technogenic areas is important and urgent due to the complexity of the instrumental study and legal requirements for estimation of potential risk factors for the ecology. Researchers of bats on the territories of wind parks are forced to rely on the international practices due to the lack of the methodological standards for collecting and analyzing the data. The main goal of the research was to study the vocal activity of bats in order to find out changes in the ecological state of animal populations potentially occurring because of the construction and operation of the wind park, and to summarize basic requirements for studying the activity of bats for the needs of the project, which relates to the construction of a wind farm. The research area is located 17 kilometers west of Mariupol, which is in Donetsk region in Ukraine. It covers the entire territory of the plant project together with the power transmission line. You see the characteristics of bat detector vantage points on the left of the slide. Here you can see the methods of gathering the data. They are ultrasound detection in separate vantage points, ultrasound detection on transets, ultrasound detection at a stationary vantage point throughout the night. One of the indicators of animal behavior is the activity index, which characterizes the theoretical possibility of registering bats over a period of time. Such calculations make it possible to characterize the periods of minimum and maximum activity of bats for further action and measurements for the management of natural environmental complexes. You can see the description for each element of the following formula on the slide. During our study, we received the following amount of signals, 190 and 173 signals in 2019 and 2020 respectively. The figure shows how the values of the activity index changes at separate vantage points. Some points showed the substantial activity. For example, the vantage point 19 over here near the village of Yalta. In our opinion, such a relatively high activity of animals is associated with the close location of the water area and settlements where bats have the opportunity to rest and forage in the lighted areas. As we can see from the following table, the activity of animals depending on the phenological phase of their annual cycle ranged from 0 signals per hour up to almost 11 signals per hour. Such values can be explained by weather conditions. According to our studies, the most dangerous period for bats is August and the first half of September. It is during that period that measurements are needed to minimize the negative impact of wind turbines on bats. We also estimated the species diversity of bats on the research territory, which is characterized by at least 10 species that belong to 6 taxonomic ranks. You can see the distribution on the following slide. None of them is being classified as an endangered species. To obtain more specific information on the territorial distribution of bats in the project area, we use the kernel density method for point objects. The result of applying this technique is the construction of zones of increased activity of bats, both in certain periods and for all observation seasons on a cartographical basis. Such zoning allows to identify places of potential risks for bats. We can see that on color schemes, and to provide appropriate recommendations on environmental management and minimization of impact. Based on our study, we created the list of impact factors for bats inhabitants in the research area. We divided them into 3 groups. First of them is the impacts caused by the construction, which includes emissions of harmful substances, deterrence by visual effects and noise, occupation of the territory by the working sites and equipment, loss of breeding sites, loss of individuals of certain species. Second group, impacts caused by equipment. This category includes long occupation of the territory and change of characteristics of the territory, deterrence by massed vertical structures, barrier effect and obstacles for flight. And the third group is impact caused by operation of the wind farm. It contains deterrence due to the rotor movement, flickering shadows, glare of light, additional development of the territory, disturbance due to the night illumination, collision with wind turbine generators. In conclusions, you can see the total number of registered voices during the research period under the number 1. Under number 2, you can see the dynamics of the index of bat activity depending on the research methodology. Forage and migratory behavior of bats, calculated by determining the index of their activity, shows the average values. The distribution of bats in the project area indicates the attraction of animals to settlements and open waters, while the wind farm area is less attractive to them. We estimated the species composition of bats. There are no species of the category endangered among the identified bats. So, based on our research, we state that the construction and operation of the wind power plant Wind Farm LLC at the level of objective forecast will not have and negative impact on the bat population of the study area. Thank you for your attention.
Thank you. Questions, please. Svetlana, please.
Thank you for your presentation. It's really interesting. I have a small question. Can you clarify for me what the period for the maximum voice activity for the bat?
Thank you very much. Thank you very much for your question. The maximum activity, voice activities of bats population is depend for the several factors. For example, we mean season of the years, we mean the phenological stage of the population, we mean the territory, it's a south part Ukraine or middle part of Ukraine or north, and it's depend from the station near the water borders. For example, you can see on the slides of our presentation the main voice activity is attraction to the water borders and settlements, for example, small village or green zone in the big town, and depends from several factors. But if you want to registrate of maximum activities, you must be going to the field in the middle part of August or early of September, because it's a period when the young generation of this year is starting for the flight, and the numbers of bats is increased, and it starts for the autumn migration when we registered the increase of species biodiversity. For example, if you want, if you registered some species, some field research in the summer period, it's only 5-7 species. But if your registration voice activity during the September, it's more than 10 species biodiversity. So answer your question, middle to the end of August and early of September is a period of maximum voice activities. Thank you.
Thank you for your answer. Small question more. And do you understand right that you can identify the species with signal detector?
You mean we used the ultrasound detectors? Yes,
yeah,
we used
this equipment, and I mean, you know which species is provide this sound. So
yeah, because we have computer programs and we analyze it of sonograms of each signal, and we have 12 parameters which we used for the identification of the species of bats. It's not completely 100% for the identification, but we have a lot of I don't know, non-problems signals, because it's very close to the species. But we have problem with identifying those species, and for example, in the group of Plecotus, it's mean it's a very similar signals between several species. So we use computer program for the analysis of the sonograms of signals.
And the terms of voice activity are the same for each of these species.
Yeah.
You see the second part of August, it's the same for each of species. Yes.
Yes, and the same.
Thank you.
Thank you. More questions? No, thank you. Please write the questions that you did not have time to ask in accordance with the regulation to the text chat. Dear colleagues, please turn on your webcams for photos regarding the report. And absent today, Oleg Punach and many other speakers, we will wait our speakers, but I am silently grateful to all the participants of the conference for their high quality research. Today we say enhance the results of studies in the field of biodiversity, ecosystem, and many other problems, which passed a double blind review and a rather difficult selection process. Once again, during our work, I already see some of our regular authors, and it makes me very happy. Thank you for your work. I am sure I am speaking for everyone when I say grateful we are to our speakers for her excellent presentation. I'd like to thank everybody here. We have come to the end of our session, so we are adjourned, after which the section. The links and the program are in the conference agenda. I sincerely thank everyone for their active participation. I hope for continued cooperation in the future. Sergey?
Thank you, Vyacheslav Volodymyrovich. It was an exciting program for your workshop. Meantime, for the first time in my practice, we have almost 10 talks, which from a single university, Bogdan Khmelnytsky Melitopol State University, which none of authors are nor registered, neither try to make some live presentation. Accordingly to our primary requirements from the EIP publishing, we must to exclude these talks from the following conference program and exclude the papers which were announced and not presented from the conference proceedings. It's a very sad case. I think it's for last time we make such a hard decision. So our obligations need to be completed. Such happens. My apologies to all session participants, and good luck. Thank you, Vyacheslav Volodymyrovich.
Thank you, Sergey Alexeyevich.